I think we were a little past this, so, but I'll start here. Okay. Nucleic acids is the last category that you need to know. Nucleic acids have a little bit of complexity. They operate to encode information in the cell in order to make proteins or to regulate other genes. So they are the instructions, like this would be one letter, right, of that ACG configuration. And it's built into three parts, okay? There's the coded part right here. Okay, this is the nitrogenous base. And if you were to look at this, there's a lot of N's here. So when you're in lab, actually, I wish I had an image like that. There's all these N's here. And so that's the best way to identify that that's the nitrogenous base. That'll definitely be in your lab. So that will be helpful there. The next part is the sugar phosphate backbone. The sugar adds the stability. The phosphate adds the reactivity. It's the glue that binds it to the next nucleotide, the next nucleotide. So we get a sequence of DNA or RNA. Okay. So even though this part doesn't code, and this is the same for every single one of these little nucleotides, this is going to be the changing part. This is the sugar phosphate backbone, right? Because it's really just about structure, not about code. All right, so things to know, at least about the DNA RNA paradigm. The sugar is deoxyribose for DNA, hence the DNA, and it's ribose for RNA. And here's all that means. It's in the name. This sugar is deoxyed. It's minus an oxygen. That's it. That is all. And it actually has a profound impact. And what it does is because we've taken ribose sugar, it's sweet, it's a little like pentamer of sugar, we've just taken off that oxygen. What's happened is it's become a tighter bonding sugar. It's more stable. And this one, ribose, allows RNA to be made quickly and then degraded quickly, and made quickly and then degraded quickly. So these messages are kind of like text messages. They're ephemeral, okay? Whereas this is supposed to last for decades, hundreds of years, like sequoia trees, thousands, right? So this is a long-term storage molecule, okay? The bases, again, are adenine, guanine, thymine, cytosine, AGTC. That's what you need to know. And then for RNA, they just have one that's a little different, and it's a modified thymine. So you still have A, you still have G, you still have C. Those are still in there. But where we have a T here, a thymine, we have a uracil. Uracil literally looks just like thymine. It just has a tiny little change, like a couple of atoms changed. Okay. But it's how you're going to tell the difference when we start doing our sequencing about it. Okay. Oftentimes, so this is in like humans. Okay. We're not talking about like viruses here. In classified living organisms, cell-based living organisms, this DNA has to last a long time. And so it's double stranded to make it extra strong, right? To double code on both ends. It's like a carbon copy. You know how, have you ever, like some people probably, you guys probably don't use this very much. There's this thing called a carbon copy. And it's like, if you write on the top sheet of paper, there's like this other sheet of paper underneath it that's kind of black, like in checkbooks. And that black is actually carbon. And so when you make the impression, it makes two copies at the same time as a carbon copy. I should never use that example ever again. It's 2024. I know what that is. You guys may not. Anyway, so that's one of the things it does. It makes a copy right at the same time. So if we have damage on one side, it's okay. We still have the original, so we can go back and fix it. Okay, so that's a really nice insurance policy built in. RNA is single-stranded again because it doesn't behoove the cell to have really strong RNA. It behooves the cell to have RNA can stick together and then take apart really fast. So there's that. Because this has to be such a strong structure, it starts turning on itself and has a three-dimensional shape, and that gives it a stronger long-term, I guess, like configuration. And I'll show you why in a little bit. RNA is just a ribbon, just a little ribbon. Or burn. Okay. All right. So nucleotides from a linear molecule are called a strand. That just means like there's all of these orders on a strand: gene A, gene B, gene C. The backbone, again, is the phosphates and the sugars. The bases are on one side. We keep it in the middle of the molecule so that they're really protected and safe. The DNA is a double helix of the 2 strands. It looks like a staircase that keeps it really solid. Hydrogen bonds are like static. You know when you have like a wool blanket and it's dark and you do that thing where you peel it up and it goes crackle, crackle, and there's all of those little sparks? That's static electricity. And so they're held together by that. And if you've ever pulled on it, you'll notice there's a little tug back, right? It bites you a little. It's not smooth, like a microfiber or something. And so that's what's holding together these helices. And even though we think of them as tiny little bonds, or yeah, I can break it. When you have a bunch of them, it's hard to break it. If you have a pencil, it's hard to break, or it's easy to break one pencil, but if you have like a whole bunch of pencils, right? Like 30 pencils, it's really hard to break them all together. And so that's the idea of hydrogen bonding. Hydrogen bonding works really well in groups because it becomes very strong when they're unified. So there's that. Here's the helical structure for DNA. And so as you can see, here's that helix. All that means is a curl. It's just a little curl, linear curl. There's bonds that they're not being represented, but they all kind of go along here. And there's hydrogen bonds in this place. Also in the middle where you see our nitrogenous bases, our code, you'll see these little dotted lines. Those are also hydrogen bonds. We call them sulfhydryl bridges, right? But essentially they're a static-like bond, okay? And so on this particular strand, DNA has some characteristics. Like A's will always bond to T's, and C's will always bond to G's. And that's something you'll have to remember. We'll revisit that, but for now, one of the ways to remember it is C and G, they kind of look like each other, so they're matching in that way, right? They both have that curve, that half moon curve, and then A and T actually form a word, and that word is at, right? You can't really rearrange anything else to do that in a 2-letter pair in that group. So it might be a little helpful. Okay. Here's a little bit more up close, right? So here's all of these little hydrogen bonds. At, or A and T, in that word, there's only 2 letters. And there's only 2 hydrogen bonds between A and T. So that might be helpful. In your lab class, they'll give you these unlabeled nucleotides. And the trick to seeing if you're dealing with an AT pair or a CG pair is really just counting these. So remember, at is a word with 2 letters in it, so it has 2 bonds. C and G, they're just the 3 bond. Okay. The last class you need to know of nucleic acids is adenosine triphosphate, ATP. It's a high energy molecule. I'll show you why. But basically what it is it's money. And if you feed it to enzymes, it will catalyze reaction. It turns them on and gives them energy. It's kind of like feeding it lots of little batteries, okay? It holds these 1, 2, 3 phosphate groups. Phosphate groups have a lot of energy. They're highly reactive. And when you stack 3 on there, if you were to hold an ATP molecule, this part where all of these phosphates are would just like buzz in your hand. It would feel like prickly almost, right? It would just have so much energy to it. And so every time we lose one of these phosphates, we burst open some energy and we can give it to an enzyme that we need to work. And that's how we use this. And it's a rechargeable. Later on, we can cap it back up. But that's an important feature of ATP. So it's adenosine, right? It's an adenine. And it still has all the things that an adenine would have, right? So it has a nitrogenous base, has the sugar, but what makes it adenosine is the fact that it's going to be bonded to instead of being bonded to a single phosphate group, it's bondable to all of these. So now it's adenosine triphosphate.